Thank you. Thank you, Peter, for the nice words, and thank you all for coming. It's really an enormous pleasure to see so many people at 9:30 in the morning in Milano. Yes, I want to talk about tuberculosis, immunity, and also some translational research, notably biomarkers, and very, very little about vaccines. So let me start with an introduction. This is someone coughing somewhere in the world. Now, chances or risk actually are high that this is the way that you can get tuberculosis. This is a very frequent event. 125,000 times a day, someone gets infected with Mycobacterium tuberculosis. This adds up to about 2 billion people who are latently infected with this pathogen. And the good news is that only about 10% during their lifetime will develop disease. Still, this makes up to 9 million cases annually, and therefore it's a major health threat, notably also because it leads to about 1.5 million deaths annually. And the situation is getting worse. First, about 15 million individuals are co-infected with HIV, and clearly HIV is the driving force for tuberculosis, and tuberculosis is the #1 cause of death in HIV-infected individuals. And second, increasing numbers of multi-resistant strains exist, with 50 million individuals being infected with multi-resistant strains. Now, this adds up to about 500,000 new cases who are drug-resistant TB, and about 1 million individuals who are co-infected with HIV and TB. And the situation is become even worse. More recently, we have heard that XDR, extensively drug-resistant TB, is arising in more than 80 countries worldwide with about, and that's a very rough estimate, 50,000 XDR TB cases. And the numbers are probably much higher because in many countries, XDR is not diagnosed. We are happy if you can diagnose TB at all, and you're not going into real diagnosis of multi-resistance. So this is XDR TB, which can still be treated, but treatment is almost impossible in many countries due to lack of financial resources, and treatment lasts for many years and not always is successful. Now, most recently, we have heard about TDR TB, which is totally resistant TB, which is clearly untreatable because there's no drug that can treat TDR, and those have been described in the 3 I's, India, Iran, and also Italy, and more recently also in South Africa. I will briefly talk a little bit in very simplified terms about immunopathology and say very, very few words about vaccines, since there's nothing really immunological new in my opinion, and this is something more for a clinical meeting. So let me look again at someone who coughs, and what happens now is that the bacilli enter the alveoli of the lung, where they are eaten up by neutrophils, alveolar macrophages, and we all know that, and perhaps most importantly also by dendritic cells, and the dendritic cells then transfer or transport the mycobacteria to the draining lymph nodes, where T cells are activated, and in healthy individuals a solid granuloma will develop, composed of CD4 T cells, CD8 T cells, macrophages, dendritic cells, a few neutrophils, and mostly then also quite a number of B cells, and it's both CD4 and CD8 T cells that take care of a solid granuloma. Once immunity weakens, for many reasons, the most typical or best known probably HIV affecting CD4 T cells, that the granuloma becomes caseous, the T cell control no longer functions, and the bacteria are spread through the alveoli into the open space, and the person is infective and other people get infected. The bacteria may also be spread to other organs, and a kind of different organ manifestations of TB can occur. So let me summarize. From an immunopathologic standpoint, 2 billion people lifelong have these solid granulomas. That might be a slight overstatement, but in principle it's true. And in 9 million people annually, the disease develops because caseous granulomas develop. So it's the transition from the solid to the caseous granuloma that is the major issue and the major distinctive factor that decides whether you're infected but healthy or you are infected and diseased. And all current vaccines, that's one of the few words I want to make, comments about vaccines, all current vaccine candidates try to stop this transition. So we don't have any vaccine candidate that is ideal, that eradicates the pathogen. All vaccine candidates currently in clinical trial are preventing the transition from a solid granuloma to a caseous granuloma, that is from infection to disease, but no sterile eradication. And that is the real challenge. Very briefly about the different vaccine candidates. First, it's MVA. These are about 12 candidates that are currently in clinical trials. In red, you see viral vectored vaccines, which express 1 or 2 or 3 antigens. This is either the modified vaccinia Ankara, developed mostly by the Oxford group, or it's the adenoviruses expressing antigens developed by Crucell or by a Canadian group. In blue, you see the vaccine candidates that are adjuvanted proteins, again, 1, 2, or 3 proteins in the appropriate adjuvant. The most developed one is the one by GSK, M72, and a number of vaccines have been also developed by the Staten Serum Institute. These are the subunit vaccines given on top of the current vaccine BCG, and then there are a few in green vaccines which are recombinant live vaccines. BCG typically, also most recently, also a vaccine that's based on a deletion mutant, double deletion mutant of Mycobacterium tuberculosis. So this year we heard about the results of the MVA trial, which had completed 2B for first efficacy, and the unfortunate news is that it did not show protective efficacy, so we have to go on. It was not better than BCG. And our vaccine, which I will not really discuss today in this talk due to time limitations, is the recombinant BCG. It has completed a phase 2A for safety and immunogenicity in newborn in South Africa. Everything looks promising, but this is no efficacy data, and we are preparing now for a phase 2B efficacy trial, most likely in South Africa and in India, because most recently the vaccine has been taken over from vaccine project management by the Serum Institute of India. Now, what is clear is that if we want to proceed with such a high number of vaccine candidates, which on the one hand is good, is a very expensive endeavor. And therefore, it would be critical to find markers that tell us today whether a vaccine works tomorrow. That's a challenging task. We are not there, but everybody in the field would agree that this needs biomarkers that allow us, first, of course, to diagnose TB in a very rapid way, and second, also to predict the development of disease. In other words, to predict whether a vaccine is efficacious or not. And I will talk a little bit about biomarkers as our translational approach to TB research. We develop biomarkers, like others, to discriminate infected individuals, the 2 billion people, from the diseased individuals, the 9 million every year. Ideally, and we are on the way to there, mostly in collaboration now with several partners in Africa, notably Willem Hannekom and Gerhard Walzel in South Africa, to predict whether one person gets disease in a couple of years. That is to say today, whether an infected individual is likely to be at risk to develop disease, predictive markers. Now, we are also, and I will focus on this today, also highly interested to use biomarkers as indicators for questions we have not asked yet, because they exist in the patient, and we can try to answer the questions in the mouse in a kind of a reverse translational approach. So our strategy is very simple. We look at the responses in the infected individuals who are healthy, the 2 billion individuals on this globe, and we compare them with those who develop serious disease, tuberculosis. That's the 9 million cases annually. And today I will really focus only on gene expression analysis, that is transcriptomics, although more recently we have done a lot, and part of this has been published on metabolites and on immunologic signatures as well. So this is a very simple summary, which is given in the title. That's an analysis of transcript biomarkers in South Africa with Gerhard Walzel from Stellenbosch University, the Gambia with Martin Ota, and Uganda with our colleagues there. And what we find indeed is that two signatures are of major interest, and these are biologic signatures that are signatures where you have a whole signal cascade. That's first the IF1 type 1 and 2 downstream signaling, and second the Fc gamma downstream signaling, and actually the role of the IFN type 1 in tuberculosis was very, very impressively shown by the paper of Nogara's group in 2010, was also seen by us and also by others. And the second one, as I said, the Fc gamma receptor downstream signaling. Now, these are biological signatures, and they're very important to better understand immunobiology, pathology underlying TB. They are less helpful in sensitivity and specificity increases for biomarkers, because if the first, in a simplified way, if the first molecule is upregulated, then the next one will follow because it's downstream of the first one. So you add less value to specificity and sensitivity. So the alternative selection clearly is to use markers that are totally independent or largely independent biologically to increase specificity and sensitivity. We've done that, and what we see really is that it's about 4 to 5 markers we can really discriminate tuberculosis patients from infected individuals, and the markers are Fc gamma receptor, lactoferrin, GBPs, guanylate binding proteins, coenzyme A, and defensins. And that's what comes out. If you take Fc gamma receptor, lactoferrin, GBP, and granzyme, 94% of our predictions have also been predicted by the clinical parameters. That is, we can predict tuberculosis and diagnose tuberculosis in 94%, and latent tuberculosis, that is infected healthy individuals, by 90%. That's quite good. And if we add then the defensins, we go over 90%, and we think that this is a high specificity and correct and robust diagnosis. So it works, but is it specific? This is the question I'm always asked by the immunological community, and we decided to go on that with a disease that's very similar to tuberculosis pathologically speaking, but is a non-infectious disease, and that's sarcoidosis. So now we do biosignatures of tuberculosis and of sarcoidosis because they are so similar pathology-wise, but they are clearly distinct by etiology. And what we see really is that the vast majority of genes that are upregulated in TB versus healthy infected individuals or healthy non-infected individuals are also upregulated in sarcoidosis. So the vast majority of all these differentially regulated genes are differentially regulated in both diseases versus healthy individuals, yet there is a lot of room also for specific upregulation versus healthiness in TB and also in sarcoidosis. And most importantly, we also find, and sometimes they are totally different genes, differentially regulated genes between tuberculosis and sarcoidosis. So what we want to propose is, and that's not so surprising for immunology actually, that the vast majority of genes that are regulated are building blocks that are upregulated or downregulated in similar diseases. That's about these 75% of the genes, and there clearly are unique genes for different diseases like tuberculosis, sarcoidosis, or we looked also at the systemic lupus. So here you find the different ones, also these are less. So what we would like to propose really is an algorithm that allows us to distinguish really first inflammation, then chronicity of disease, immunocompetent versus immune diseases, and so on. That is not a single step saying this is the signature of TB versus everything, but rather a small signature that distinguishes first diseases versus healthiness and then go down to the specific disease. Now, these were long-lasting endeavors which took a lot of time over the last 5 to 8 years. And the original philosophy was really we come from the bench, go to the bed, and then to the bush in TB. And more recently, we clearly realized that this is only one way at looking at it, and rather than going only this way, we increasingly now going way back because we see so many genes where we know they are important, but we don't know why they are important. That is, we don't fully understand their biological function, and therefore we are now doing a lot of reverse translation, which I will spend a little bit in the next couple of minutes. I call that reverse translation, and I would like to mention from the beginning on that often with these markers, the distinguishing feature, the distinctive feature really is that these are not yes or no phenomena, but rather these are balanced issues which you cannot really study in humans, but rather needs the mouse, because it's a balance where often too much is bad and too little is bad, and the right balance correct. Or saying it with Paracelsus, it's the dose that makes the poison, dosis sola facit venenum. So let me start actually with interferon type 1. And before I do that, just 1 second again, we will focus now mostly on monocytes, macrophages, and neutrophils. And we will focus really on chemokines, since we found that many of the phenomena we are studying are really due to differential regulation of chemokines in the lung at the rather early phase of tuberculosis. Let's start with type 1. As I said, we already know from Enogara studies, from our studies, that type 1 IFN is a distinctive signature in TB patients. We know really no clear answer about the role of type 1 IFN in TB. That's still controversial. So our decision was to take a highly TB susceptible mouse. You know, many mouse strains are quite resistant to TB, and that's the 129S2 mouse, a substrain of the 129 family, and we compare them first with B6 mice. And then later we also introduce a knockout mouse on that background deficient in the IFN type 1 signaling, and you will see that all the TB susceptibility in these mice is related to the IFN1 signaling. So this is his first experiment where you see that the wild type mouse really dies by day 30. And if you knock out the type 1 IFN, and if you knock out the type 1 interferon, you will see that you rescue all the mice. You see that by lethality, you see it by colony-forming units, and you will also see that the pathology is quite markedly different with enormously large granulomatous lesions in the wild type mice, the 129 mice. What's the reason for that? We first looked at cytokines and chemokines, and we see indeed that in all cases, the 129 mice have elevated levels of pro-inflammatory cytokines, IL-1, TNF, IL-6, also in GCSF, and also in the chemokines, CXCL1, CCL3, CCL2. These are the ones that we could easily measure by multiplexing in the lung. So upregulation of these pro-inflammatory cytokines and notably also chemokines seems to be a distinctive feature of these mice. What are the consequences? Well, what we see is that very early, already at day 14, these mice have elevated levels, slightly elevated levels of neutrophils in the lung at day 14, which gets down to almost normal level in the IFN knockout mice. And we see also CXCL1 and CXCL5, two of the major chemokines that regulate migration of neutrophils into the lung, upregulated. This is very clear at day 21 when we have a huge abundance of neutrophils in the lung and much lower abundance in the IFN knockout mice. And this is clearly on the expense of alveolar macrophages where you don't see that. Does this have any consequences? Now, we look at neutrophils or macrophages now and look at their bacterial burden. So the very rapid influx of neutrophils, we look now whether they are able to kill the mycobacteria or whether they maintain the mycobacteria. And what we see is that the early neutrophils indeed fail to kill the mycobacteria and therefore seem to promote mycobacterial load. I think that this is contrary to what most people would have assumed, at least what I would have assumed. So can we rescue these mice? Yes, we can. If we neutralize the neutrophils, that is, we kill the neutrophils, indeed these mice are rescued, as you see on this slide. So we transform a susceptible phenotype into a resistant phenotype. So let me summarize briefly the first one of these kind of studies. The 120 mice are highly susceptible, and type 1 IFN deletion really rescues these mice. The 129 mice are really susceptible, at least correlated, I would argue, even due to high increase of CXL, chemokines, and PMN influx, reduced MTB control in these neutrophils, and exacerbated pathology. And finally, we can rescue indeed the mice by depleting the neutrophils. In a very simplified scheme, in these 129 mice, you have a lot of neutrophils migrating into the lung alveoli. This leads to damage. If you rescue these mice by IFN receptor or IFN type 1 signaling deletion, then you get a normal composition in the alveolar space, and you can rescue and help these mice to survive longer against TB. Next one is microRNA 223 as a typical example. This comes from our studies where we compare tuberculosis with sarcoidosis and where we looked not only on the transcripts of the messenger RNA, but also on microRNA, and found that indeed a few microRNAs are differentially regulated in TB, tuberculosis patients, and in healthy individuals infected. We are not the only ones who see that, and one of the microRNAs that are differentially regulated is microRNA 223. They have been described in Asian population. We saw it in a Caucasian population here in Europe, actually in Italy, and it's also been seen by us in African population. So it seems that it is one of the differentially regulated markers amongst the microRNAs. And this is indicated here in peripheral blood, where you see upregulation of microRNA 223 in TB patients as compared to latently infected individuals. And perhaps more importantly, we see that also in lesions. So if you take a healthy lesion and look at a sample there and a diseased lesion and compare infected region versus non-infected region, and therefore we see also a marked upregulation in the lung of patients suffering of TB. So what's known about microRNA 223? Indeed, it is known for long that they are preferentially expressed in myeloid lineage and have been mostly thought to control the differentiation and maturation of neutrophils and mononuclear phagocytes. In fact, they have been shown to be negative regulators of neutrophils. That is, neutrophil microRNA deficiency leads to exacerbated disease in defense against Candida. And finally, more recently, a control through NF-kappa B has been described. So what we decided to do is now look first as a susceptible mouse and compare that with a resistant mouse to see whether in the lung of these mice, susceptible mice versus resistant mice, we see a differential regulation of microRNA 223. And indeed, that is the case at day 25, where we compare healthy tissue versus diseased tissue. And you see this upregulation of microRNA 223 in the susceptible 129 mice, which I just described to you, and no upregulation at day 25 in the resistant mouse strain. So what about knockout mice? These mice exist, and what we see indeed is that in these mice, when you delete microRNA 223, they are much more susceptible to tuberculosis, as indicated here, and again you also see increased pathology in these mice. So microRNA 223, although it's upregulated in diseased patients, is something that seems to be helpful, but of course again we keep in mind that this is often a matter of balance rather than a yes or no phenomenon. So we predicted potential targets of microRNA 223 on the mRNA level, and what we find indeed is that CXCL2, IL-6, and CCL3 pop up as interesting candidates predicted as direct targets of microRNA 223, and indeed all three of them are upregulated both on the messenger RNA level and on the protein level. Now, if you look at the regulation by NF-kappa B of these three cytokines, chemokines, you will see that they are all under direct control of NF-kappa B. So the question comes up, is this now an effect that goes through NF-kappa B, or is it indeed direct modulation of these three mediators? Now, the evidence we have is that they are direct targets of microRNA. If you take a reporter cell line, which either expresses CXCL2, IL-6, or CCL3 at the microRNA and the appropriate controls, you will see downregulation of all three, of IL-6, CXCL2, and of CL3. That is, we consider this quite experimental proof that the prediction was right, that these three molecules are indeed direct targets of microRNA 223. Can we rescue these mice again? Yes, we can, and it's quite dramatic. We can rescue them by neutralizing or depleting the neutrophils with a single shot of neutrophil, anti-neutrophil antibodies, only partially, I confess. Second, we can rescue them by a combination of antibodies that neutralize the three mediators, CXCL2, CCL3, IL6. And finally, we can most dramatically rescue them by using a double knockout mouse of CXCR2 and microRNA 223. So now CXCR2 is the receptor for CXCL3 and, of course, many other chemokines, and I come back to that. So let me summarize here. microRNA 223 indeed controls migration of the neutrophils. It orchestrates this migration by directly targeting CXCL2 and other molecules. And through this, microRNA 223 again is a kind of balancing factor in the early events in tuberculosis. And again, in this cartoon, what we see here is the microRNA is regulating a kind of homeostatic system. If you delete it, you get increased influx, notably of neutrophils, and if you then delete, and therefore you get a lot of damage, and if you then delete also the CXCR2 receptor, which is the receptor for the ligands like CXCL3, which are involved in that migration, you can rescue the mice again. The last example actually builds up on these findings, namely that the CXCR2 is a central mediator in this particular situation. And the question we raised here actually is the CXCR2 is a receptor for a multitude of chemokines, including CXCL1, 2, 3, 5, and 15. And the question was, is this really a totally redundant story? Is this really a redundant event? Or can the deletion of a single chemokine affect tuberculosis? And I try to convince you, if we delete the CXCL5, one of the ligands for CXCR2, that's sufficient to see a major effect. So despite the shared receptor usage, the assumed redundancy, you see differences probably due to spatiotemporal differences between the different chemokines, so they are produced at different stages. And this is done, starts here with the looking at the transcripts in a mouse under TB infection, low-dose infection with MTB, high-dose infection of MTB. And if you look at the different CXL chemokines shown here and many other molecules, you see differential regulation. But clearly, the CXCL5 is the most prominent one. It appears very early, it gets very high during high-dose tuberculosis infection. So if you look, I have to make that very clear, at low-dose M tuberculosis infection, 50 to 100 bacteria per aerosol, effects can be seen, but they are minimal. If you take a higher dose of MTB, then you mimic a situation where you already have some pathologic events being induced very rapidly. Under these circumstances, you see indeed that the CXCL5 depletion rescues these mice. Many of the mice, 50% of the mice, die under these high dose in the first 40 to 50 days of infection, and you can rescue these mice by deleting a single chemokine, despite all the redundancy and shared receptor usage. And you also see again that neutrophil influx is a decisive factor, because in the CXCL5 mice, neutrophils are really lower, whereas in a high-dose infection of TB, there's a massive influx of the neutrophils. This is markedly reduced by deletion of the single chemokine. And the question then was, which cells are the producers? Well, CXCL5 indeed is produced rapidly, and it's produced most rapidly by those cells that are the first to see Mycobacterium tuberculosis, that is the epithelial cells of the lung. So these cells are the producers of CXCL5, neither the macrophages nor the neutrophils produces. That's different to the CXCL2, which is produced mostly by the myeloid cells, that is by macrophages and neutrophils, and it's different to CXCL1, which is produced by both cell types, both by the myeloid cells, the macrophages mostly, and by the epithelial cells, which are in contact rapidly. So this brings in the epithelial cells into the sensing of Mycobacterium tuberculosis at a very early stage, and therefore in protection and pathology and early orchestration of immunity against tuberculosis. What are the molecules that are recognized? How are MTB recognized? The most likely and major factor is toll-like receptor 2. We see activation by whole mycobacterial cell wall components. We see also activation by lipoarabinomannan, lipomannans, the 19-KD lipoprotein, and that's equally high to the controls like the PEM3, which works also through TLR3, or the LPS, which, as we all know, works through the TLR4. And we can block all the mycobacterial activities by blocking TLR2. These are in vitro studies, but still they indicate that indeed mycobacteria are rapidly seen by the TLR2, sensed by the TLR2, which activates then the production of chemokines, notably CXCL5 in the epithelial cells. So MTB induces CXCL5 in epithelial cells through TLR2. CXCL5 knockout mice show increased resistance to TB, and increased resistance is related against to reduce neutrophil recruitment and activation. We consider it important that despite this shared receptor usage, there's a single chemokine that does quite a lot of effects, and therefore there is not just redundancy, and we consider it most likely that this is due to spatiotemporal differences between the different chemokines, the CXCL5 being produced earlier and more profoundly, very rapidly after infection. And again, this is consistent with the detrimental role of neutrophils in tuberculosis. And again, let me look at this kind of scheme. What we see here again is a low-dose TB infection where there is everything in order, a kind of balance. And at high-dose infection, neutrophils enter the lung alveoli. We see a lot of damage. We see a lot of damage in the alveolar space due to the enormous production of CXCL5 and other chemokines, but still it's sufficient then to delete this single chemokine to re-establish a kind of homeostatic event during the tuberculosis, and the recognition of the mycobacteria in this case is really due to recognition via TLR2 by the epithelial type 2 cells in the alveoli, which are the main producers of the CXCL5. And so we have a situation where a radio-resistant, non-immunologic epithelial cell really starts these effects in tuberculosis. So in all these cases, we saw that there are differences depending on the dose of the, better say, the concentration of the molecules, a balance between type 1 IFN, a balance in the microRNA 223, and also a balance in the chemokines, which are important, of course, to attract the phagocytes to the lung, but at higher abundance cause damage. And in all our cases, the neutrophils were really a culprit, a major part of the pathologic responses. And therefore, I would agree again with Paracelsus that it's the dose that makes the poison, or perhaps better, it makes the dose or concentration that makes the damage. Let me end by thanking the people who did the work. Anka Dorhoy, who's in the audience, and Geraldine Nouet did all major work together with Vlad Yeremev, who's now in Moscow, and Marco Iannacone from Naples, Italy, on the basic research, or in our case, the reverse translational research. Martin Gengenbacher and Steve Fries are the ones who drive our vaccine efforts forward. We have a next generation of vaccine, but I didn't talk about that. And Jaron Matzdorf, January Weiner, Mark Jacobsen, who has now left to Düsseldorf in Germany, are the driving forces in the biomarker research. And we do this, of course, not by ourselves alone, but in strong collaborations. The microRNA 223 mice were a kind gift from Fernando Camargo. Leander Grode and Bernd Eiseler from VPM bring forward the clinical trials of our vaccine candidate, and Mark Cotton is the one who is responsible for the clinical trials in South Africa. And our biomarker studies are supported by the Bill and Melinda Gates Foundation in a huge consortium, the GC6 consortium. I can't mention all of them, but I will mention the major players: Gerhard Walzel, Martin Otter, Willem Hanekom, and Harriet Mayanja, and all the others. And the sarcoidosis studies were done with the German clinician Joachim Müller-Quernheim. We receive a lot of funding from the Max Planck Society, from the Gates Foundation, the German Ministry for Science and Technology, the German Science Foundation, and also from the NIH. And we are located in the middle of Berlin, just opposite of the Bundestag, which is our government center. If you are coming to this lively city, let us know and visit us, and thank you for your attention.